So, thank you very much for inviting me. I'm an academic lecturer in Liverpool with a clinical and research interest in epilepsy. Today I'm presenting my pilot study which used links, data, and tools to find drugs that could be repurposed for the treatment of epilepsy. I'll start by giving an introduction to epilepsy, how epilepsy is treated, shortcomings of current anti-epileptic drugs, and why better anti-epileptic drugs are needed. I'll then present my pilot study and planned work that builds upon the pilot study. So I know that the audience is primarily composed of data scientists creating links, data, and tools. So first of all, thank you very much for providing this great resource. I hope that this talk will be able to add to your understanding of how clinicians and biologists are taking advantage of these opportunities created by links to find therapeutic leads within their disease areas of interest. And I would be very grateful for any suggestions from you on how to make best use of links in my work. So epilepsy is defined as a disorder of the brain characterized by an enduring predisposition to generate epileptic seizures. 1% of the population, or 65 million people worldwide, have epilepsy. And the global disease burden from epilepsy is higher than that for Alzheimer's disease, multiple sclerosis, and Parkinson's disease combined. At the individual level, epilepsy is associated with increased risk of death and disability, psychological, social, and financial harm. Epilepsy can be treated with anti-epileptic drugs. There is no shortage of anti-epileptic drugs. This long list shows many, but not all of the currently available anti-epileptic drugs. But however, these drugs have significant shortcomings. So currently available anti-epileptic drugs act primarily upon a small number of targets involved mainly in modulating neuronal excitability. Hence, these drugs suppress seizures but have no impact on the underlying pathophysiology of epilepsy. So in other words, these drugs don't cure epilepsy, they only stop it from manifesting as seizures while the individual continues to take the drugs. However, they even fail to achieve seizure suppression in 30% of people with epilepsy, even when taken at maximal doses and in combinations. Over 100 years of using traditional drug discovery approaches and the development of more than 30 anti-epileptic drugs has failed to overcome these challenges. But potentially, these challenges could be overcome by utilizing genomics-based drug repurposing. We know that hundreds of genes influence susceptibility to epilepsy and are dysregulated in the epileptic focus. Drugs identified by genomics-based drug repurposing will target multiple genes dysregulated in the epileptic focus, thereby restoring the global gene expression patterns to their healthy norm, and as a result, correcting the diverse pathological changes that create and promote the hyperexcitable neuronal network which generates seizures. So this approach offers the potential not only of improved anti-ictogenesis, i.e. suppressing seizures, but also of anti-epileptogenesis, i.e. cure or disease modification. So our aim is to identify new anti-epileptic drugs using genomics-based drug repurposing. So for the transcriptomic signature of disease, we used a mouse model of epilepsy. There are many different mouse models. From these, we chose to use the pilocarpine model, as gene co-expression networks are conserved between this mouse model of epilepsy and human epilepsy. The transcriptomic signature was extracted from a published RNA sequencing analysis. Differential expression was defined using conventional criteria, and our disease signature consisted of unordered sets of up and down regulated genes. The pattern matching or connectivity mapping against links drug signatures was performed using the links cloud query tool, which of course has now been replaced by L1000 CDS squared and the Clue app. The mean of the 4 highest connectivity scores was used as recommended by the app, and scores with a magnitude greater than 85 were considered significant. This led to the identification of 123 compounds which met the mean connectivity score criteria. It was noteworthy, but not surprising, that this drug set contained no conventional licensed antiepileptic drugs, as we know that conventional antiepileptic drugs act primarily on a small number of targets which modulate neuronal excitability. So, in other words, our results suggest that the repurposable compounds identified are better than conventional antiepileptic drugs at reversing the transcriptomic dysregulation underlying epilepsy. But we wish to corroborate this using a different paradigm and an independent database of drug signatures. So we asked the question, are repurposable drugs better than conventional antiepileptic drugs at reversing the biological pathway dysregulation underlying epilepsy? To answer this question, we used the drug set enrichment analysis, or DSEA data, which has been generated by the Di Bernardino group in Italy. This data set was created with a different function in mind, but we repurposed it, if you will, for our own needs. So DSEA includes biological pathways from multiple databases, for example, Reactome. And each pathway in the database is stored as a ranked list of drugs, sorted from the one most upregulating the pathway to the one most downregulating it. So for any pathway up or downregulated in disease, the DSEA dataset can be used for identifying the compound most effective at reversing the dysregulation. And this dataset is based upon CMAP 2.0 dataset. So this is the strategy that we used. Reactome pathways dysregulated in the epileptic focus were identified by performing gene set enrichment analysis on the results of the previously mentioned RNA sequencing study. The 100 most upregulated and the 100 most downregulated pathways were noted, and then this strategy was adopted. In turn, I compared each repurposable drug with the set of conventional antiepileptic drugs included in DSEA. For each dysregulated pathway, I determined if the repurposable drug is better than conventional drugs at rectifying the dysregulation. And finally, I determined if the repurposable drug outperforms conventional drugs for more pathways than expected by chance alone, based on the exact binomial test. So this bar chart shows representative results from the comparison between one of the repurposable drugs in red and all the conventional antiepileptic drugs in blue. On the vertical axis is the percentage of dysregulated pathways corrected most effectively by each drug. And as you can see, the repurposable drug is the best performing compound for the highest proportion of pathways. The fraction of dysregulated pathways for which the repurposable drug surpassed conventional antiepileptic drugs was statistically significant. So this data is for one of the repurposable drugs called camptothecin, and this analysis was repeated for an unselected sample of repurposable drugs and produced similar results for each of them. So while there were no conventional antiepileptic drugs amongst the repurposable drugs identified, I was interested in finding out if any compounds had efficacy in rodent models of epilepsy. Rodent models of epilepsy are a vital tool in antiepileptic drug development. Efficacy of drugs in rodent models is generally predictive of their effectiveness in people with epilepsy. These models are currently the gold standard in vivo validation tools for potential antiepileptic drugs. A brief literature search suggested that some of the compounds did indeed have efficacy in rodent models of epilepsy, which prompted me to determine if the repurposable drugs were significantly enriched with drugs of known efficacy in rodent models. So to determine this, we adopted the following strategy. Each of the roughly 20,000 compounds in the links database at that time were annotated as either having published evidence of efficacy in animal models of epilepsy or lacking such evidence. The number of compounds with evidence of efficacy amongst the set of 123 repurposable drugs was determined. Then the linked compounds were divided into random sets of the same size as the repurposable set, and the number of drugs with efficacy was determined for each random set. The number of random drug sets with the same or greater number of efficacious drugs as the repurposable drug set was determined. So we have to start by annotating each of the roughly 20,000 compounds as either having published evidence of efficacy in animal models of epilepsy or lacking such evidence. Clearly, this was a challenging text mining task, and this was achieved by mining data from Medline abstracts. Our strategy was based on the observation that sentence-level concurrence of search terms in Medline abstracts increases specificity and precision of data retrieval. So abstracts were downloaded from Medline. They were then parsed using Unix command-line text manipulation tools. The abstracts were broken into individual sentences. A sentence was parsed if the following terms concurred within it. Number one, the name of a compound from the links database. Number two, the term suppresses or a synonym thereof. Number three, the term seizures or a synonym thereof. And finally, the name of a recognized rodent model of epilepsy. The results were manually reviewed to exclude irrelevant results. For example, if a sentence read, metformin suppresses seizures in the pilocarpine model of epilepsy, that sentence would be parsed, and that was taken as evidence of the efficacy of metformin. This analysis showed that the repurposable drugs were six-fold more enriched with drugs having published evidence of antiepileptic efficacy in rodent models than expected by chance alone. This enrichment was statistically significant both by permutation-based analysis and according to the hypergeometric distribution. And to the best of our knowledge, this is the first time a quantitative estimate of the accuracy of genomics-based repurposing predictions for a specific disease has been shown. Next, we wanted to study the most promising drugs in more detail. So we filtered out drugs that carried high risk of toxicity at therapeutic doses, that required parenteral administration, that lacked data to indicate an effective dose, or were blood-brain barrier impermeable according to published literature. After this process, 36 compounds were retained. We performed a more in-depth literature review of these 36 drugs. We found that many of the drugs have compelling evidence of efficacy in multiple animal models of epilepsy, some of which is shown here. And in fact, a number of the drugs have published evidence of anti-epileptogenic efficacy models. In other words, the potential for disease modification or cure. Some examples being shown on this slide here. Next, we chose a drug to perform our own in vivo validation. We wanted to choose a drug that was not previously studied in animal models of epilepsy and had MHRA and EMA approval. These are the UK and European equivalents of the FDA, I believe. And the drug also should have extensive data of routine, safe clinical use in humans, as such a drug would encounter the fewest research and regulatory barriers on the repurposing pathway and will be more readily adopted by the clinical community. After excluding drugs that already have published evidence of efficacy in animal models of epilepsy, sitagliptin was the highest ranked MHRA EMA approved drug in routine clinical use in the UK. Sitagliptin is a commonly used oral medication for diabetes. The model we chose to test this drug in is the 6 Hz psychomotor seizure model. This is a model of drug-resistant epilepsy. Seizures in this model are resistant to many established anti-epileptic drugs, for example, carbamazepine, ethosuximide, lamotrigine, phenytoin, and topiramate. Sitagliptin and vehicle control were administered intraperitoneally. Mice were then given rectangular currents via corneal electrodes, and the seizure scores were recorded for each animal. And we found that in this model of drug-resistant epilepsy, sitagliptin produces a dose-dependent reduction in seizures, which is statistically significant at the highest dose tested. So those were the main findings from our pilot study. And I'll move on now to our research plans, which build upon these results. So our immediate plans include further in vivo validation analyses using sitagliptin and other identified repurposable compounds in rodent models of acute seizures and chronic epilepsy. We are also planning an analysis of electronic health record data to determine if the use of repurposable drugs by people with epilepsy for indications other than epilepsy is associated with improved seizure control. Looking to the longer term plans, we are very keen to take repurposable drugs for the treatment of epilepsy all the way from bench to bedside. We have particular expertise in anti-epileptic drug trials, which will be vital, of course, in this process. For example, the multi-center standard study, which is the largest ever anti-epileptic drug trial, was performed by our department. However, in our view, this pilot study has revealed the potential of genomics-based repurposing for identifying new anti-epileptic drugs. But before investing significant resources in clinical trials, we need to maximize this potential through a systematic program study that optimizes and customizes each step of the genomics-based repurposing process for anti-epileptic drug discovery. So in our view, this will involve addressing a number of questions. For example, can the predictions of potential anti-epileptic drugs be improved by using the transcriptomic profile of epileptic human hippocampi as opposed to rodent hippocampi, or by formulating disease signatures using more stringent criteria for differential expression, or by using drug signatures generated only from neuronally relevant cell lines, or by using alternative pattern matching algorithms, or other important methodological variations. We plan to answer these questions using empirical evidence relevant to epilepsy by testing each important methodological variation, thereby generating multiple sets of candidate drugs. From amongst these, the best drug set will be identified quantitatively by comparing enrichment with compounds of known anti-epileptic efficacy, as done in this pilot study. We will then take the top drugs from the best approach onto in vivo validation, then clinical trials, and then hopefully into the clinic. We feel that this systematic approach could be emulated for other diseases as well. So to conclude, genomics-based repurposing identifies a set of compounds significantly enriched with drugs possessing efficacy in animal models of epilepsy, and hence it is an appealing methodology for the discovery of potential anti-epileptic drugs. The potential efficacy of repurposable drugs against drug-resistant epilepsy and epileptogenesis warrants further study. The methodology used to quantitatively estimate the accuracy of repurposing predictions could be generalizable to other diseases, and we are in the process of creating some user-friendly scripts for performing this function. And finally, we plan to create a customized and optimized bioinformatics approach for genomics-based repurposing in epilepsy in order to maximize the chances of finding effective anti-epileptic and anti-epileptogenic drugs. So I'll finish off by thanking colleagues who were involved in this work, Graham Sills, Munir P. Mohammed, and Tony Marson. If there are any questions, I'd be happy to answer. Can you hear me?
Yeah, I believe we can hear you well. So does anybody have a question? So can I ask you, what is the overlap between the animal model tested drugs and the ones that you designated as potentially repurposable?
So in the ones that we felt were potentially repurposable, there were so there were out of the 36, there were 10 that already had published evidence of antiepileptic efficacy or disease-modifying efficacy. And in addition to that, we tested sitagliptin, which was not tested before, and found that to be efficacious as well. So from the ones that we had shortlisted, it's a encouragingly high hit rate, we think.
I'm just trying to figure out what was your 2x2 table when you actually tested these Fisher's, right, enrichment or overrepresentation of the drug.
Yes. So let me just go back to that. So our basic strategy was essentially a permutation-based approach. So we annotated each of the compounds in the database as either having efficacy or lacking efficacy. Based on this evidence, we then annotated our 123 repurposable drugs which had met the inclusion, which had met the connectivity score criteria. So we annotated those drugs as having efficacy or lacking efficacy. Then we divided the whole of the database into 123 random sets and determined if any of the sets had the same number or more efficacious drugs as the repurposable drug set. So that was the permutation-based approach. Then for the hypergeometric distribution approach, or obviously you could apply a Fisher test to this. Hopefully I'll be able to explain this verbally without any visual aid. So the boxes be the 123 repurposable drugs, and how many of them have evidence of efficacy in animal models, and in the next row all the drugs in the database and how many of those have efficacy in animal models. I hope I've explained that reasonably.
Yeah, I get that direction actually. But so how many? So the overlap was that were tested and showed efficacious was 10. Did you say that out of 123?
So the 10 were. The 10 I was referring to was from a more in-depth analysis of the more promising drugs.
Okay, so
from this I can't remember off the top of my head exactly how many were overlapped. So they were perhaps a bit more than 10. Out of the whole. Go ahead.
Okay, no, I guess, so it doesn't really matter the exact time. I'm kind of wondering what's your space of negatives, right? Because you have certain number of them tested in animal models. But for the negative ones, these were not tested, or there was no data on them being tested?
Yeah, well, exactly. So it may be that they were tested and they were found to be ineffective, but for the majority of them, they hadn't been They hadn't been tested. So we don't really know, for many of them they haven't been tested, so we don't know whether they, yeah, so we don't have that data for many of them.
Okay.
Yeah, but they're surprisingly large. I mean, we were surprised, but there's a surprisingly large number of drugs which have been tested in animal models of epilepsy, and a surprisingly large number of drugs which actually have evidence of being effective in animal models. And it's probably a reflection of the fact that animal models of epilepsy are reasonably easy to utilize and interpret, perhaps compared to other brain diseases or other diseases where the animal models might be more difficult to deal with.
All right, thank you. I was just wondering, the orders of magnitude of how many were actually tested with animals, because that seems to be a kind of really nice background, right, to have validation set, to have. Anyway, thank you. Sorry.
It seems like someone entered a question in the chat. I don't know if you can actually see it.
Oh, I see. Let me see. Let me click. All right. Yeah. So the only the only other medication that I'm aware of sitagliptin being used for is. So I'm sorry. Can everybody see that question, or should I read that out?
Well, maybe you can read it, or I can read it. What do you want me to read it? So it's basically Dr. Mirza does the best effective dose of, I can't read this because it's too small. Sitagliptin also influence other diseases. In other words, is the dose effective for epilepsy alone?
Yeah. So the only other I'm aware of sitagliptin being used for is diabetes, where it's been a licensed drug for over a decade. So the doses, the most effective the most effective dose in our study, which was statistically significant, is somewhat higher than the one that is routinely used for the treatment of diabetes. But it is the dose is at a level which has been used previously in animal models without ill effect, and there have also been reports of human use at that level without ill effect. So yeah, so it's a bit higher than is currently licensed, at least in the UK. Obviously, we're converting the rodent dose to the human dose. It's a bit higher than is currently licensed, at least in the UK, but is likely to be a safe dose.
So it seems that the most effective dose in vivo experiments which you performed, it was 500 milligrams per kilogram, right?
Yes.
That's, I mean, seems like awfully a big dose. And I guess the question would be, did you test any other anti-diabetic drugs sort of to see, is it, phenomenon of lowering blood sugar? I mean, sort of, or is it the sitagliptin specific effect?
So we haven't the short answer to that question. I mean that's an interesting question. What we can say is that in people who are not diabetic, sitagliptin does not cause hypoglycemia. So it wouldn't be expected to cause hypoglycemia based on that, based on that information. But I accept that that's a good question. What effect is it might be having on glucose homeostasis, and could that be relevant? So that's something for future study, but it's not something that we have looked at.
the follow-up question is actually, one of the effects of sitagliptin side effects actually is inflamed pancreas. Did you guys look, since, and the human dose is about usually 100 milligrams daily, oral administration. So you guys intraperitoneally injected 500 milligrams per kilogram. So did you guys look at any organs, particular pancreas of the mice?
So we didn't do our study, but they in previous studies in literature, these kind of levels have been used without obvious problems or kind of pancreatic problems, etc. So because there was some data on this already, we haven't. We didn't study that specifically when we were doing the study.
So I have a question for you. So there's many different forms of epilepsy, and so I guess I was wondering if you could talk a little bit more about how you identified the pathways that you think should be corrected in order to treat epilepsy, given the variety, and more specifically, I guess, which form of epilepsy do you think is most amenable to this approach?
Thank you. That's a very good question. So the mouse model that we used is a model of the medial temporal lobe epilepsies. So partial or focal onset epilepsies where there's sclerosis of the hippocampus. So these findings are based on that. These findings are most likely to be of relevance to that form of epilepsy. This form of epilepsy is the most common cause of drug-resistant epilepsy. So it is a useful type of epilepsy to start off with targeting. Whether these drugs, if they're found to be effective for this model, whether they'll be applicable to other forms of epilepsy is something that would have to be looked at. Certainly there are animal models of other forms of epilepsy as well, and in future studies, one thing to look at would be to test these drugs in other models as well, which model other forms of epilepsy, to see if they are effective in those models as well.
Thank you all for attending, and we'll see you in the next iteration of the CIC data science webinar.